Welcome, everyone. My name is Renee Duprel, and I'm the Director of Development here at the Institute for Systems Biology. Thank you all for joining us for our Research Roundtable. This is our virtual series that we host to keep you up to date on research at ISB. This series is geared towards non-scientists, and our goal is to make our work more approachable to all. Our intention is to have one of these just about every month, so keep an eye out for our emails. Today, we'll be hearing from our featured scientist, and then there will be an opportunity for questions and answers after that. Those of you who join us regularly know that once in a while we have a giveaway, and we have one today. Megan O'Rourke is a well-known, let's make sure you see that, is a well-known writer for the likes of the Atlantic Monthly, the New York Times, New Yorker, and more. She has a new book out, The Invisible Kingdom, Reimagining Chronic Illness. Megan will be in conversation with ISB President Jim Heath at Town Hall Seattle on Wednesday, September 14th, and we'd love to see you all there. That will be a virtual event too, just like this. Just by joining us today, you're entered to win the book, and we'll let you know at the end of the hour who that lucky person is. Today's topic is bugs versus drugs, how our unique gut microbiomes shape our personalized responses to statins. There's a Q&A box at the bottom of your screen, and you're welcome to submit questions at any time. I'll be monitoring those, and at the end of the presentation, we'll move to the question and answer session, and we'll work through those questions. Now I have the pleasure of introducing Sean Gibbons. Sean is an assistant professor here at ISB and the Washington Research Foundation Distinguished Investigator. Sean was born and raised in Montana and went to the University of Montana for his undergraduate studies. Sean has 3 bachelor's degrees, microbiology, molecular biology, and French literature. Sean finished his PhD in biophysics at the University of Chicago in 2015, and he started his faculty position here at ISB in June of 2018. Welcome, Sean.
Thanks, Renee, and thanks to ISB for letting me share my science with you today. Let me see if I can get my screen going here. All right. So yeah, thanks for turning out, everybody. Today, I'm going to discuss some very recent work that has come out of the group in the past couple of months, where we tackle a really interesting problem in the microbiome field, which is, how does our unique gut bacterial composition affect the ways in which we respond to various pharmaceutical drugs? So I'll just jump right in. And I guess before I run too far, I want to call out this really beautiful piece of art as a part of my cover slide here that Allison Kudla here at ISB put together for us. We tried to pitch it as the cover art for the journal Med, where this paper was published. Sadly, they chose a different cover, but I'm presenting it to you all so you can kind of see what it would have looked like. But basically, yeah. How does our microbiome affect the ways in which we respond to drugs, and in particular, statins? If you've heard me give a talk before, I'm not going to spend too much time introducing the microbiome, other than to say that we are not alone. Our bodies are home to a very diverse array of microorganisms, trillions upon trillions of cells in and on our bodies. Everywhere that's exposed to the outside world is just covered in bacteria, whether that's our gut or our skin or our mouth or our nose. And over the course of our evolution, we've actually outsourced some of the functionality of our bodies to this complicated ecosystem that inhabits our bodies. Indeed, even if you look at the genetic capacity of the human genome versus the microbiota, there's hundreds of times more genes in our microbiome than there is in our genomes. More than 99% of the genetic capacity that you can find in and on our bodies is pent up in our microbiota. We know it's important, but we're just beginning to decipher exactly under what conditions it's important and how it's important. Obviously, in order to study this kind of a thing, you have to take a systems approach. You have to measure various parts of the body, both what's happening inside the host, and what's happening with the microbes, and then try to decipher or understand what amount of the variation, what amount of the change in what's going on in the microbial ecology of your bodies actually impacts, your health and your responses to interventions. So I think, the microbiome is at this point pretty widely considered a new organ of the body, right? Not a classical organ. It's got a mind of its own, right? It's this diverse set of species that are very different from us and have their own goals and things they want to do. But essentially, many of the metabolites that circulate throughout our bodies and our bloodstream are derived from our gut bugs, and it has enormous number of effects on all the systems of the body. This is just one little PDF that the journal Cell put out a while back. Sort of gives a cartoon view of what's known about how the microbiome affects the body, but you don't need to read it all to see that pretty much every organ system of the human body has been associated with the microbiome. So we know that it has wide-ranging effects. This sort of tendrils are reaching into all the parts of the body. And the question that we're here going to ask today is, do they affect our responses to pharmaceuticals, to drugs? Does the variability in how different people respond to the same exact drug, whether it be aspirin or a prescription drug like a statin, does your microbiota affect those responses, mediate those responses somehow? And we're going to focus on statins. And why will we focus on statins? The main reason is that they are the most commonly prescribed pharmaceutical, I think, on planet Earth, but definitely in the developed world, they're the most common drug taken by people. Over 20% of all Americans over 40 years old are taking statins, 1 in 5, and it's growing over time. There's many different types of statins, but the big ones on the market right now in the US are Lipitor and Zocor or Flolipid. And essentially, the reason we take these drugs is because they reduce our risk of heart attack and stroke. So they're widely used as a preventative measure to decrease the risk of heart disease and stroke in people. Now, statins save lives. They have been widely shown to have a very positive effect on human lifespan and the prevention of heart attacks and strokes. And that is huge, right? It was a big win for medicine, just like antibiotics were a big win. Statins were also a big win. But similar to antibiotics, it's not all good. Even though you probably should be taking a statin if you're at high risk of heart attack, independent of any other concerns, you probably should be taking it. Nonetheless, there is a bit of a side effect on your metabolism. So LDL, lipids, and sort of glucose homeostasis, these things are sort of coupled together. And if you mess with your lipid synthesis in the body, you can kind of make your metabolism a little wonky. And it's been observed that people who are on statins are at a higher risk for transitioning into type 2 diabetes. So that's what this plot is showing. You take a matched cohort of people that are balanced for their age and their sex and other risk factors, and you follow them over time. People who are taking statins tend to transition to diabetes at a higher frequency over the intervening years than someone who's not on a statin. And recent work has come out to show that people who are already diabetic and are given statins, and indeed diabetic patients are actually targeted for statin treatment because they are themselves at much higher risk of heart attack. So cardiologists will actually target diabetics to receive high dose statin treatments. But this recent study showed that you could measure the fact that if you gave someone statins, that they progressed more rapidly in their diabetes. Now, you could argue that it's a lesser of two evils, right? Would you rather die of a heart attack in 2 years, or would you rather have long-term side effects due to diabetes over the course of decades? That's something to consider. So I'm not trying to scare anybody away from taking statins. Statins are good. And if your cardiologist says take a statin, take a statin. But I'm trying to maybe open our minds to a new world in which we empower people to have more control over how they're treated, and that perhaps we can find a balance between side effects and protection. So we know statins' effects on human populations are really, really heterogeneous. You can see in this plot here, This is the amount of LDL change in a large population of folks, about 8,000 people, all given the same dose of statin over the same period of time. And some people actually saw a rise in their LDL cholesterol in that window, right? And some people showed a huge drop, almost 100% drop in their LDL. And on average, people brought their LDL down by about 50%. And that 50% is what the drug company puts on their advertising, right? That's the clinical target for this drug, and they hit that target. But you can see that there's no such thing as an average human, right? There's a few people that are at 50%, but much of the population is on the ends of this distribution. This trial also showed that the extent to which you can bring down your LDL is associated significantly with the extent to which you have events in terms of heart attacks. So the more you bring down LDL, the less likely you are to have a heart attack. And so this was sort of the impetus for what we wanted to look at in this Aryville cohort that we have access to, is looking across this large population. Can we explain some of this heterogeneity using deep phenotypic information, and in particular through looking at the microbiome? And I should call out my collaborators here. This work was driven by Tomasz Romański, who's a very talented research scientist in Nathan Price and Lee Hood's labs, who has since moved on to Amgen, and he drove this work. And my co-corresponding authors, Noah Rappaport and Andrew Magis, were sort of co-mentoring on this paper, and Christian, who's in my lab, helped with data wrangling and processing the microbiome data. So it was a really big team effort and team lift to get this work out. Now, when you embark on a study, you have to look at what other people have done before you. To remind you, our interest was really, does the microbiome modulate how people respond to a drug? But there was a study a couple of years ago where they asked a slightly different question, which is, do statins modify the ecology of the microbiome? So are statins causally changing the composition of the gut microbiome? And this was in Nature a couple of years back, and this is a European group, and in Europe they have this method for clustering things into what are called enterotypes. That's what these colors are, and I'll explain that a bit more later. Basically, they cluster people into groupings that seem to share similar signatures as far as what organisms are present in their microbiomes. And so there's the names of them are BAC1, BAC2, PREV, and Rumenaccaceae, Prevotella, Bacteroides, and Rumenaccaceae. And I don't think you need to know much about this particular plot, except for the fact that the BAC2 group is considered less healthy. It tends to be enriched in people who are obese or who have diabetes or other issues. And indeed, if you look at this plot on the bottom here, as you increase BMI, you see that the fraction of people with this BAC2 enterotype generally rises with higher and higher BMIs. But the big punchline of this paper is when they looked at people who are on statins, they found a sort of marginally significant effect where statin users who were obese tended to not have this BAC2 enterotype. So they maybe were protected from this dysbiotic or bad microbial state. And that was the sort of main punchline of this particular paper. You'll see later why, I'll throw a wrench into that a little bit, that result. But that's one question you can ask is, do statins affect the microbes rather than does microbes affect the response to statins? Another reason we would look at this problem is there have been a few studies recently that have looked at the capacity of our gut microbiome to degrade or transform a wide pool of available pharmaceuticals. So for hundreds of pharmaceutical drugs tested, the gut microbiome seems to be able to break down something like 20 to 30% of these compounds. And among them are statins. So this is a figure from that paper where you can see individual bacterial taxonomic names on the x-axis here, colored by their phylum level annotation. And the bars are showing the amount of drug consumed in a sort of in vitro assay. So, this is, for example, Bacteroides uniformis is a particular species of microbe that lives in the human gut, and it consumed about 50% of lovastatin when it was incubated with the drug in vitro. So we know that the bugs in our guts can break these things down. They can transform or break down these statins, and that could be affecting the responsiveness of us, how much of the drug is making its way into the bloodstream and having its effect on our bodies could be affected by these types of metabolisms. And generally speaking, one observation that's come out of this work is that this particular phylum, Bacteroides, is more enriched, it seems, for these kinds of drug metabolisms. So, I've been sort of thinking about, what is my working model or my mechanistic model for how all this fits together and how all this works. And I'll kind of dump some of that knowledge on you to have floating around in the backs of your minds while you take in these results. You have a couple of observations where the correlation between dietary cholesterol intake and circulating cholesterol is actually not that great. It doesn't seem that the amount of cholesterol you eat really affects the amount of cholesterol in your blood a ton. And in fact, it's actually high carbohydrate diets or simple carbohydrate sugar diets that are more likely to increase your cholesterol levels. So eating sugar actually affects your cholesterol more than eating cholesterol. And this is maybe because of the fact that more than half of the cholesterol that's in your body right now is produced by you. It's produced by your liver. So the biggest source of cholesterol in the body is coming from host metabolism. And so that's what statins target. So statins are these drugs, originally, I believe, isolated from fungi that inhibit a particular enzyme in our bodies. It's the rate limiting step in the synthesis of cholesterol. It's called HMG-CoA reductase. And so that's the direct effect that statins have on us, is it gets into the blood and goes to the liver, and it affects this enzyme and reduces the amount of cholesterol that's produced in our bodies. Now, how could the microbiome be mediating all of this? One direct way is that if statins are acting as either a nutrient source or as a poison to the microbes, they could be eating that statin, degrading it either as a source of energy or to get rid of it because it's toxic. A sort of similar vein here is the microbes, even independent of their sort of growth or toxicity, they could still be breaking down or modulating these drugs and turning them into either even more potent versions of the drug, like a pro-drug, so to speak, or they could be degrading them into an inactive form, sort of interfering directly with HMG here, with this, which is CoA reductase inhibitor. And then the last piece here is the microbes have all kinds of effects on other systems of the body. Microbes can actually transform cholesterol into a molecule called caprostanol. And caprostanol cannot be reabsorbed back in through the gut. And so if cholesterol in the gut is turned into caprostanol, it's a one-way ticket for pooping. It's going to be lost by defecation. So the more your microbiome is converting cholesterol into caprostanol, more of the cholesterol in the actual lumen of the intestine is going to be lost from the body. Microbes can affect bile acid metabolism, and bile acids are derived from cholesterol, and their levels can affect various receptors in the body and affect the synthesis, the pathways for synthesizing cholesterol and the various derivatives of cholesterol. And there's also been interesting work showing that the mucus layer in our guts, the amount of consumption of that mucus layer, the barrier integrity and degradation of mucus can potentially affect whether drugs and things can pass through into the bloodstream. So lots of potential mechanisms. And so we have to kind of look now into the blood. We have to kind of look both at the stool and the microbes and look at what's passing in through the blood and compare the two and try to understand what's going on. So now we're getting to the meat of the actual study. We looked at the Araville population. Many of you are familiar with this population of people, thousands of people where we have really dense information about them. We've sampled their blood and their poop. We have metabolomics, proteomics, microbiomes, and we can kind of compare across the stool and the blood. And for this particular subset of the population, about 1,800 or so people, we had reliable information on whether or not they were taking statins. So 1,600 people were not consuming statins, and 244 were on statins in the Arabelle population. Of the people that were known statin users, for a subset of them, less than half of them, we actually had quantitative information on the dosing of statins they were receiving. So whether they were low, moderate, or high intensity doses of statins. And how are we measuring the effect of these statins? The classic effect of a statin, the way we measure it is through the change in LDL cholesterol over time after you start taking the drug. Sadly, with Arabelle, with this particular analysis, we only had one time point to look at here for most people. And so we had a snapshot. We couldn't calculate that change. So we came up with another kind of way to validate our results and look at efficacy by looking at the buildup of this molecule HMG, which, if you remember, HMG-CoA reductase is the enzyme that's being inhibited by statins. So you get a buildup of this HMG-CoA, and the esterase will cleave the CoA off, and this HMG molecule will build up in the blood. And so we can measure this as a sort of on-target efficacy. The higher this HMG level in the blood, the more statins should be inhibiting the enzyme, ostensibly. And if we look in the blood of people who are or are not taking statins, we kind of get a sanity check on that, on what I just told you. So in non-users, you see there's no correlation between plasma HMG levels and LDL cholesterol in the blood. But as soon as you start taking statins, and those who aren't on statins, you see this negative association, where the higher HMG is, the more it's building up. Ostensibly, the more the enzyme is being inhibited, the lower your LDL cholesterol is pushed down. And this is significantly so. And generally speaking, people tend to be shifted towards higher HMG levels if they're taking statins. This is also true for dose. This effect of HMG as an effect, as a measure of an effect, can be seen across dosages. So people who are taking higher and higher doses of statins see higher and higher, progressively higher levels of this HMG in their blood. And there's a large variation as well, which is what we're interested in. We're interested in the variability of the effect. And this is also associated with LDL. There's another way we can look at this, which is there are known genetic markers for your responsiveness to statins, right? Genomically, some people are known to be better responders to statins than others. And so by we, I mean Tom, pulled down, I think 9 of the most sort of powerful or strongest hits for these markers from various genomic surveys that have been done. And of those, this particular variant I'm plotting here in the ApoE gene is the strongest effect modulator for statin effect. And when we look at plasma HMG in non-users and statin users, we see that there's no difference between the different genotypes in people who are not taking statins. But as soon as people take statins, first of all, you see that everybody has a jump upward in their HMG, which means the drug is working. But the people with this particular allele, this particular variant of the gene, the T allele, they see a bigger hop upward in HMG, which means a bigger effect, which is exactly what we expect to see. This is what has been reported previously. So this HMG seems to be a good marker of on-target efficacy. So what if we look at it in terms of the microbes in the gut? So one of the simplest things you can look at is the diversity of the microbiome. How many species are there, or the evenness of the abundance distributions of species in the ecosystem. And what I'm showing here, again, I'm using these little cartoons to show you where the data is coming from. So the x-axis is coming from the microbiome, which is poop, and the y-axis is this HMG in the blood. And we've split the diversity of microbiomes into 3 different groupings, tertiles, so to speak. So low diversity, intermediate diversity, and high diversity as you go from left to right. Blue are people who are not taking statins, and red are people who are taking statins. And what you can see is that people with the lowest diversity microbiomes see the biggest jump upward in their HMG, which means that low diversity microbiomes show stronger effects of the statin. They have higher effects from statins than people with higher diversity microbiomes, which is sort of puzzling and kind of interesting. You could ask, is this due to varying dosages across people who have varying diversities? And the answer is no. So for the people where we have dosing information, we can adjust our models and control for dose, and we see that the effect persists. There's this negative association between your effect of a statin and how diverse your microbiome is. Ostensibly, one way to explain this is that if you have more species available, the chances that you have a particular microbe that has the genetic capacity to transform a statin goes up. And so maybe in higher diversity communities, the capacity of the microbiome to metabolize statins increases, and maybe that's what's driving the effect. But we don't know if that's the case or not. That's just one way of thinking about it. So we can look beyond diversity. And since there was this prior paper that looked at these things called enterotypes, which are clusters, kind of coarse-grained clusters of microbiomes, we did the same sort of analysis in the ERBL data set. So Tom clustered the ERBL participants into these enterotypes, and luckily, this is true in developed countries, essentially, like US and Europe. You tend to see that people cluster into these 4 groupings. 2 of them, BAC1 and BAC2, are enriched for this particular genus Bacteroides, which is a common genus that's found in the gut microbiome. This green group, Prevotella, is enriched for a particular bug called Prevotella, which sort of is abundant when Bacteroides is less abundant. And it's very clearly associated with this cluster, right? You see a huge jump in Prevotella in this group. And then the BAC2 group, what really distinguishes it from the other groupings is that it tends to be depleted in bacteria that are thought to be good guys, generally speaking, butyrate-producing Clostridia. One of them is Faecalibacterium prausnitzii, which is the last plot there. And you see that BAC2 is most depleted on average in that particular organism. And so these results kind of matched what we saw before in that other paper that was published previously. So can we replicate what they found, right? They found that there was a protective effect of using statins, so that if you were obese, you were actually less likely to be in this back to potentially dysbiotic or bad grouping. Well, we had kind of a wah-wah on that. We did not find it. In fact, we saw the polar opposite. So people who were taking statins who had a BMI of 30 or higher actually were enriched for this BAC2 group. We were actually able to replicate this in another independent cohort. And we took the same data set that prior group had analyzed, and the only way we could replicate what they found was by restricting our analysis to people with advanced heart disease. So I don't know what that means, right? Like only with people with advanced heart disease show the effect that they report. It could be a survival bias, right? Like if you have this unhealthy microbiome that's associated with diabetes and obesity, perhaps you're less likely to survive when you're in that group. And so if you also have heart problems, maybe you're just not capturing those people because they die and you don't sample them. So I don't know what's going on there. But anyway, this is sort of a tangent because really what we found is there isn't really much of an effect of statins on the microbiome. Statins don't seem to have a huge effect in modulating the composition of the microbes. Their effect actually seems to be much greater in modifying the responsiveness of individuals to drugs. So then zooming into these enterotypes again and looking at HMG as a sort of a measure of that effect, we see variability between these groups. Generally speaking, the people who are enriched in Bacteroides, they have a bigger effect. They get a bigger bump upwards compared to the same people with the same enterotype who aren't taking statins. They get a bigger bump upwards than everybody else. And in particular, the BAC2 group gets the biggest bump. It also happens to be the group with the lowest diversity. So that could be, I think that's largely what's driving the diversity signal I showed you before, is that group is just low diversity in general. But almost all the enterotypes show a significant increase in HMG when you take statins, including this Rominococcaceae group, just not as much. Prevotella didn't show a significant effect, but you don't really find that many Prevotella folks in the developed world. So there were only 13 people on statins with the high Prevotella category. So I take that with a grain of salt because we don't really have the numbers to do a really robust statistical analysis on that particular group. If you look at the amount of LDL cholesterol reduction across these enterotypes, there's pretty dramatic differences. Back-2 really drops your LDL down. Like for the same statin bang, you get more buck, essentially, when it comes to bringing down that LDL. Now, what's interesting on the side effect side of things is if you look at markers of prediabetes or diabetes, you find that those same groups that had the biggest effect for lowering their LDL and reducing their heart disease risk, they also had the biggest perturbation to their metabolism. So the back-2 group in particular showed the biggest difference between matched controls, not on statins, in this particular marker, HOMO-IR, which is a marker of insulin resistance. But you can look at lots of other markers of metabolic perturbation, like HbA1c or glucose or insulin, and they were all perturbed in the same direction. And for both these Bacteroides groups, they were metabolically perturbed seemingly by taking statins. I should also caveat that this is a cross-sectional study, so we weren't looking at people over time, so we can't necessarily say there's a causality, but we did our best to control for confounding variables. And down at the bottom of the screen, I often list all the covariates. So we're controlling for age and BMI, sex, other batch effects, and other physiological parameters. So we think it's a robust sort of observation. Now, one of the coolest things we see here is this Rominococcaceae group seems to be protected from this perturbation in their metabolism. They can take statins, but they don't have the side effect when it comes to their HOMO-IR or HbA1c. So seemingly that group could probably get a higher dose of statin and be totally fine metabolically speaking. Whereas the other Bacteroides groups, maybe if you reduced their dose, you're still going to achieve good LDL lowering sort of responses while perhaps mitigating this metabolic perturbation. We could replicate this result in a completely independent European cohort. It's a cohort called Metacardis that was recruited across multiple countries in Europe to study heart disease and the microbiome. And these plots are just showing some validations, right? So this is the abundance of Bacteroides and its association with both a marker of prediabetes or insulin resistance, HbA1c here. So there's a significant association between that bug and that marker in the blood, only in people on statins, and same is true for HMG. And these associations disappear in those who are not taking statins. The enterotype associations we found, again, exactly recapitulated in this cohort. So we think that this is a really robust and reproducible observation. So I'm nearing the end here, just giving some takeaways. This is the graphical abstract for our paper in Med that was published a couple of months ago. We think what we really want to do next. We see this really cool effect, right? We see that there are variations in responses to this drug, and that variation seemingly is explained by heterogeneity in the composition of the gut. The ecology of the gut is explaining some of the variability in this response. In addition to that, I should mention, I haven't shown this data, but we did the analysis. If you look at the genetic markers of statin responsiveness, we find that if you build a model with all those genetic markers, you can explain a fairly large proportion of the variation. If you build a model with the microbes, you can explain another proportion of the variation. If you build a model with both in them, you essentially double your amount of variance explained. It's additive, so that there's not overlapping variance being explained by those two features. So it seems like whatever information you're getting from genetics is independent of whatever information you're getting from your microbes. And you can use them potentially together to better inform maybe precision statin treatment in the future. Now, even though we've seen this effect, and we think it's really interesting and worth following up on, we don't know the mechanisms involved yet. And so we are pursuing routes towards trying to better understand what exactly is the mechanism, how exactly are the microbes modifying this host response. Ultimately, we would love to run a human trial, either observational to follow people over time when they go onto a statin for the first time and see how they respond and track their genome and their microbiome. And intervention would be interesting as well. Based on the information we have, we could potentially design precision dosing regimes for people based on both their genetics and their microbiomes, and we could maybe see if a precision treatment did better than the sort of standard of care treatment that someone would receive if they walked into a hospital. Coming back to our mechanistic view of how are things working, we've learned a few things. So I think we kind of don't believe that the statins are really affecting the composition of the microbiome all that much. They do a little bit, and it's very subtle, but we don't think that that's the major way in which there's an effect of statins being mediated. We do think that there's potentially still degradation of statins or conversion of statins into a prodrug that could be occurring by the microbiota. And that's actually something that Christian Diener got funded for recently in an innovator award to do in vitro assays to ask whether or not you can measure this effect in the lab, taking poop from different people and adding statin to that poop and incubating it and seeing if statin's being degraded, if its effect on the enzyme is being modulated. But I think for exploring some of the other mechanisms, we really would need to either go into a mouse model or do a human trial of some kind. But I think we're making real progress. I think, this is sort of one of the first proofs of concept, I would say, for, precision pharmaceutical treatment that uses the microbiome information to sort of guide what you might do. People have shown that microbes can degrade drugs, but we're one of the first examples where we've shown in vivo that there seemingly is an effect of modulation or mediation by the microbiome in how we respond to a drug. So with that, I will end. I thank all my wonderful lab members. The summer researchers we have in the lab right now have been great. The funders, Washington Research Foundation, especially for my startup package. And recently we got some big grants from the National Institutes of Health, particular NIDDK and this Kennedy Shriver National Institute of Child Health and Human Development. And thanks to all of you for listening, and I'm happy to take any questions.
Oops. Great, thanks, Sean. So we do have some questions. And this first one doesn't have so much to do with statins, but that's okay. It has to do with the microbiome. And I know you can answer any of those questions too. When you take a supplemental probiotic, why do you need to continually take them? Why don't they survive in our bodies? Are they necessary for our bodies?
That's a big question, and it's a complicated answer. I'll try to go fast. So there are lots of ways probiotics can be used. The classical probiotics are limited in what they are. They're limited by what's generally regarded as safe by the Food and Drug Administration. There's a list of organisms that you can give to humans that have been checked as safe. A lot of them are just present in food already. They're already in yogurt or they're in sauerkraut or what have you. And so they get grandfathered in as safe because they're food components. A lot of the bugs that are dominant in the human gut, the anaerobes that live in the gut, I couldn't actually give them to you. It would be illegal for me to give you a pill of the Calibacterium presnitzii, even though most of us are carrying it around inside of us right now. It's not permitted by the FDA for me to do that. So the probiotics that would probably be the most efficacious, I cannot even give you. Now that's one thing. Another thing is if you give 10 people the same probiotic, in a few of them, it might stick and persist in them after they stop taking the probiotic, and in the rest, it might wash out. And if you look in their microbiomes, you'll see that there's a hole in their niche space that fits that particular organism for the people where it sticks. And for the people where it doesn't stick, there's already another organism living in that hole that is sort of competing against that organism. So you have to sort of sequence someone's microbiome before you know what organisms might engraft and sort of predict that engraftment. And finally, you can cook a lot of probiotics, kill them, pasteurize them, and give them to people, and you can still get an effect. So there's a bug called Akkermansia muciniphila that they sell a pasteurized form of it as a probiotic, and it's like taking a drug. It reduces inflammation. It's been shown in clinical trials to reduce inflammation, but it's dead. It's just dead cells, and it has an effect on you.
Okay, that is more complicated than I thought your answer was going to be, but accurate, I'm sure. Okay, next question. Is there any current research regarding the effect of statins on small intestinal bacterial overgrowth?
Not that I know of. The biggest work on that is with proton pump inhibitors, and in there, yes, definitely, right. If you're taking proton pump inhibitors, your pH goes up in the small intestinal compartment, and there you can easily get overgrowth of bacteria. The human body pretty much tries to kill everything in the stomach and in the small intestine. We want to have premier access to our diet, so we keep the pH really low. We secrete a bunch of antimicrobial peptides to reduce and kill off most of our bugs in that compartment. And in the colon, we sort of let off the reins and we let the bugs grow. As far as statins affecting that sort of host control over bacterial growth, I can't think of how they would do that, honestly.
Okay, thank you. Does an increase of HMG in your body affect your body in any way? And please remind us what HMG is in your response, Sean.
Oh my gosh, I am not going to remember the actual long drawn out chemical name. That is HMG. You can Wikipedia it at HMG-CoA reductase or HMG, and you'll find the full name on the internet. Sorry if I don't know the name.
That's okay.
Whether it higher or lower levels influence your health, that's a really good question. I don't think anyone's really studied it. No one really has really used it before as a marker of statin efficacy. I think we're maybe one of the first groups to leverage that particular molecule for that application. Although it's known that it's the precursor for the formation of that metabolic, which is the next step in the progression of the synthesis pathway that enzyme that's being inhibited by statins carries out. So it makes sense that it's building up. We see that people who are older have higher levels of HMG. So age is associated with progressively more building up of HMG, and of course, age is also associated with progressively, higher LDL and perturbed metabolism.
Okay, great. How much statins, for example, the percentage of what's taken can actually end up in the gut and interact with the microbes?
Yeah, that's a really good question, and I don't totally know the answer to it. Right there, the small intestine is probably where a lot of statin drug gets absorbed into the blood, but we can see it in poop. We've done metabolomics, and we see that in stool we can measure detectable levels of statins, and we can measure the microbes sort of eating them or degrading them. We can see its concentration going down over time. If we add nutrients to the stool homogenate, they will break down the statins. So some fraction of the statins making its way into the colon, but how much exactly, I don't know if anyone knows that. Okay.
If I wanted to know the makeup of my gut microbiome, how would I go about doing that? Is there a service or services that you know of and that are reputable?
There are lots of commercial services that will sequence your microbiome. I don't want to preferentially call out too many company names, but yeah, if you Google microbiome test, maybe I'll mention like a few so I hedge my bets. There's actually kind of an academic way you can maybe get it done. There's a group at University of California, San Diego. What are they called? It used to be the American Gut, and now it's the Microsetta Initiative. And they used to provide microbiome sequencing to the public. I think they might be in a hiatus right now, but they probably will start up doing it again. And that's an academic way of going. There's also commercial. So my old colleague, Nathan Price, actually works at this company now called Thorne Health Tech. And they provide metagenomic sequencing service for your microbiome.
Oh, okay.
A lot of these companies, they provide the sequencing and then they provide advice. Yeah. I often tell people the advice is sort of preliminary still, right? Like a lot of it is based on sort of scant evidence and correlation. But if you just want to get your data and look at what microbes are in your gut, a lot of these services are equivalent. They'll essentially sequence it and give you that information. Ideally, look for services that will give you back the sort of raw data. I guess maybe many of you don't care about that, but myself, I would really want to have the actual data. They usually just give you a profile of like what's abundant and what their names
Well,
are.
you know what to do with the data. That's the catch, isn't it?
Totally, but just like 23andMe, right? You can download your own genomic data and you can upload it to other services to get different types of analyses done on that data. If you can have your data, you can at least in the future give it to other services to get other information out of it.
Okay, got it. Okay, is it possible to increase microbiome diversity over the course of your lifetime?
Yes, easy. Eat more veggies and fruits and fiber. If you increase fiber content, you will, in weeks to months, increase the diversity of your gut.
I think I've increased my bean intake because of you. So
great.
Thank you for that.
You probably have a more diverse gut microbiome.
There we go. Okay. Let's see. I'm trying to.
Another way to increase your diversity is to be constipated, but I don't suggest you use that particular route.
Really?
Yeah, I think everything is contextual. High diversity is not always good. People with parasites have higher diversity. People who are constipated have higher diversity. You want the kind of higher diversity that's associated with complicated, diverse, vegetable and plant intake.
Right.
You don't want the kind of diversity that's associated with constipation.
Okay. Okay. Where are statins absorbed from the GI tract? If they are absorbed in the small intestine, then they would have minimal contact with the fecal microbiome.
Yeah, totally. And that goes back to the question that was already posed, right? It's true that a fair amount of the statin is likely absorbed in the small intestine, but we definitely see it showing up in the stool.
Okay. Do you think that this experimental setup could be replicated to try and study the effects of antibiotics on the gut? What might some challenges in doing that be?
Well, there are many, many studies of antibiotics and their effects on the gut microbiome, both in non-human animal models and in humans, in the context of disease or in the context of health. So those experiments have already been done.
Okay.
Well, I think what's exciting to me is looking beyond statins. The reason we focused on statins in particular, I mean, antibiotics, I should say, are designed to kill microbes, right? So we already know the mechanisms on there, and that's sort of well understood to some extent.
Yeah.
Statins, we didn't really a priori have a strong reason to think that they might be mediated in how they affect the body through the composition of the microbes. So it's sort of surprising that we see this. And I suspect that the same will be true for many other types of drugs, right? Antidepressants or proton pump inhibitors and any number of other drugs. It would be very interesting to do the same kind of analysis and just see are people who are strong responders or weak responders, can we explain that heterogeneity through the composition of the microbiome or the genome or both, or bringing them all together. And that builds out a sort of web of our understanding for how we might use that, genomics and microbiome to develop precision pharmaceuticals.
Okay.
Yeah, that's the goal.
Okay. Another side effect of statins is muscle atrophy. Do you think targeting the microbiome could potentially reduce the side effect?
Muscle atrophy. I know about myopathies and pain, like muscle pain. I'm not an expert in the clinical side of all of this, so atrophy is a little new to me. I mean, I know muscle atrophy is also commonly associated with just aging in general, but pain for sure. So a lot of people who take statins, like maybe 5 or 10%, experience a significant degree of muscle pain, and there are genomic markers of that. There are particular mutations in the human genome that are associated with worse or less bad muscle pain in response to statins. So you can kind of predict it. Whether or not the microbiome is associated with that is hard to say because we didn't have the information. We didn't have data on that kind of a side effect, but it would be interesting to look at. I mean, ultimately, though, if you are able to dose statins in a more precision manner, it means that maybe for some people, you give them more statin, but for some people, you can give them less. And for people who suffer from these kinds of side effects, taking a lower intensity statin would probably mediate or alleviate those side effects.
Okay.
How can you find a tolerable dose for someone?
Right.
Right now, it's just trial and error.
Yeah.
But we had a rational way to decide before you even give somebody the pill what they should get and avoid all the pain of that trial and error.
Yeah. Yeah. Right. Right. How can we confirm if these differences between statin efficacy and pre-diabetic markers was the result of impact of statin on microbiome health or a result of microbiome metabolism of statins? Do you want me to read that again?
I think I get it. I think I
Okay.
understand. Ultimately, like I said, we don't know the mechanism. And I suspect that the mechanism is actually pretty indirect for some of what's going on here. Like, I think there's a lot of reason to believe that the on-target responsiveness to these statins is being mediated through secondary bile acid metabolism by the gut microbiome. So yeah, I mean, for example, when it comes to the metabolic side effects of statins, one of the signatures we see in the microbiome is the Ruminocaceae group is protected, seemingly from these side effects. That group, Firmicutes in general, contains a lot of these butyrate-producing Clostridia that are known to have positive effects on metabolism. They're people with more of these bugs are less likely to suffer from things like diabetes or prediabetes or metabolic syndrome. Akkermansia muciniphila, which I mentioned earlier in the probiotic conversation, it was also enriched in people who had less side effect, or conversely depleted in people who had more metabolic side effect. And this bug is also in clinical trials as a probiotic been shown to improve symptoms of prediabetes. So you could imagine that what our results are pointing to are even co-therapies where you would not only give someone the drug, but you could maybe give them a protective microbiome intervention that would shield them from the consequences of taking the drug. So you could give them Akkermansia muciniphila as a probiotic that would maybe have a positive mediating influence on their metabolism while giving them the statin. There's just a lot to learn still. We know there's an effect going on here. We can measure it, we can see it, but what are the causal arrows and the tendrils that are affecting it all? I'm guessing there's a lot of things involved, and we just need to piece it apart still.
Okay. Okay, last question. Did you see in New Zealand, a first patient has gene editing to lower LDL cholesterol? Any thoughts on statins versus gene editing?
Oh, that's really interesting. I had not heard that.
Yeah.
That seems like a really radical intervention to reduce this like risk factor that's not going to kill you tomorrow, but maybe
okay.
Right? It's like I wouldn't volunteer for such a therapy.
Yeah.
Maybe there's some like genetic background where LDL is like crazy high and it's going to kill you
Yeah.
if you're a teenager or something. Perhaps that's the case. I guess what I would say is when we look at the genetics and we look at the microbiome, they both explain variation in responsiveness to statins and variation in LDL and HMG, but that the variance explained is completely separate. So whatever the genome is explaining seems to be separate and apart from what the microbiome is explaining. So even if you were to CRISPR someone's genome to affect their genomic responsiveness to a statin to make them more responsive on the genomic side, you could get even a bigger boost by modulating the microbiome, right? That you can affect the effect of the microbiome through the genome, it seems. They both have independent effects on the system, at least in this case.
Okay. Great. Well, thank you, Sean. Thanks for your time today.
Of course, thank you.
Yeah. Okay, so just letting everybody, thank you all for showing up too. We so appreciate you being here. There will be no research roundtable in August, but we do have a number of events coming up we'd like to tell you about. As we mentioned on September 14th, Jim Heath, the ISB president, will be at Town Hall Seattle. That's a virtual event, and he'll be talking to Megan O'Rourke, the author of this book. Registration is not open for that yet, and we'll keep you posted. You should hopefully be on our email list. If you joined us today, we have your email. On Thursday, September 29th is our annual Reimagine event, which is, it raises much needed funds for our research here at ISB. The theme this year is Reimagine Healthcare. It's going to be really interesting. We'll be showing how our research makes it from the lab all the way to the doctors and the nurses and the patients on the front lines of treatment through our partnership with Providence Health. And on Thursday, November 10th is our annual fundraiser for our STEM education events. I know we're going out really far, but we want you to mark your calendars. We'd love to see you there. We will be having kind of a happy hour style event right here at ISB. So you can come, you're going to be able to tour a lab. You'll have a chance to experience some of the curriculum that our partner schools are using around health and wellness and disease prevention. So it should be really great. You'll even learn something. Okay, it's time to give away our book. The winner of our book is Carolyn Bryant. Congratulations, you have some extra summer reading. And if you're on our email list, you'll get more information about all those events I just talked about. Thank you again for coming. And if you're inspired to make a gift to our research, you can go to isbscience.org and click on the donate button. Thank you very much, and we'll see you this fall. Goodbye.